front. Okay, this is Anabina. And so what you'll do, one person per team of 4, we've got slides and cover slips up here. So if one person per team will make a wet mount, you can actually look at the living Anabina. Now, it might be that there's more than Anabina in here. The Anabina that we saw yesterday were forming these beautiful chains of cells, so very thin, very delicate. And instead of being round like the nose stalk, which is what I was expecting, they're more rectangular. So you see these delicate little green hair-like filaments of these chains of Anabina. Beautiful stuff. Okay, right. So that finishes our prokaryotes. So then we'll flip over to page 59, your eukaryotic microbes and other organisms. So we're going cellular eukaryotes, domain eukarya. We have 2 examples of members of kingdom Protista. And the first one listed on page 59 is Amoeba proteus. Where are you going to find Amoeba proteus, you guys? Where are you going to find the slide? Micro B box slide 24, dynamite. Okay, Amoeba proteus. It's an animal-like protista. We call it a protozoan. It's free living. It's free living. This one doesn't cause disease. The second member of kingdom Protista is the algae Spirogyra. And Spirogyra, we have prepared slides up front on the tray. So you come up and get a prepared slide and look at that. And in addition, we have living cultures of Spirogyra. So again, we'd ask one person per team of 4. We've got several trays of Anabina. Several trays of Spirogyra. We've got one tray here, another one here, and another one here. We've got microscope slides and cover slips. So you're going to make a wet mount of Spirogyra and take a look at these beautiful algae. They too are going to form beautiful filaments, chains of cells. And what's going to knock your eyes out, you guys, Spirogyra has this beautiful green helical or spiral chloroplast. And what's cool, yesterday we were noticing that the size of the spiral chloroplast in Spirogyra, so let's see if I can't, they're much bigger than the cyanobacteria here. They're much bigger than the cyanobacteria, and they're going to form chains. And what's beautiful is they have this spiral chloroplast. And what folks were noticing was the size of the spiral chloroplast. It was the same size as the Anabina. And it was like, that is so cool because, remember, according to the endosymbiotic theory, where did those chloroplasts evolve from?
Cyanobacteria.
Yeah, they evolved from the cyanobacteria. So it was so cool to see that the size of the Anabina cyanobacteria looked really similar to the size of those spiral chloroplasts. It was really fun. In one wet mount, the folks had both Spirogyra and they had the Anabina, and it was just so cool. It was so cool seeing the difference in size between the eukaryotic algae and the prokaryotic cyanobacterium, and then seeing how that little cyanobacteria was just the right size for that spiral chloroplast. It was really fun. Okay. Continuing with domain eukarya on page 59, the next 2 are Saccharomyces Cerevisiae. This is brewer's or baker's yeast. This is a yeast we use to make leavened bread, and also it's the yeast that folks use to take grape juice and convert it into wine, or to take hops and grains and convert them into beer. So Saccharomyces is a unicellular fungus. We call unicellular fungi, we call them yeasts. So you look at a prepared stain smear of Saccharomyces. Now be careful, you guys, these guys are really little. I mean, they're bigger than the bacteria, but they're pretty little. They're little oval cells. And what's fun is that they can undergo asexual reproduction in a process called budding. And it'll look like one of the cells, it'll look like it's growing a nose. That's called a bud. This is an example of asymmetric cytokinesis or mitosis. And eventually that little bud, it'll actually separate and grow into a large cell itself. So you'll see in some of the Saccharomyces them undergoing this process called budding. which is typical of yeasts. The second member of kingdom fungi is Rhizopus. This is a filamentous multicellular fungus that we call molds. The common name for Rhizopus is bread mold. We often see it growing on old bread. Now, the heads up here, you guys, Rhizopus, like many fungi, can reproduce asexually or sexually. So the two slides you're looking at show you, one of the slides is showing asexual reproduction in Rhizopus. The second slide shows sexual reproduction, and they look very different. So just be aware you aren't going crazy. Okay, that's them carrying out. There are two different types of reproduction. Just two more to go, you guys, on my page 60. We're moved to members of kingdom Animalia. You'll be looking at a helminth or a worm. This is Taenia Solium, the fork tapeworm. And what you're examining is a immature worm that forms a cyst in muscle, the muscle of pigs, muscle of pork. Okay? And this immature worm, this immature Taenia, is called a cysticercus. Now they're quite big. So I would recommend that instead of using your light microscopes, use one of the dissecting scopes. These will slightly magnify the cysticercus, but it will give you a better idea of the entire structure. If you use your light microscopes, you're just going to see a little portion of the cysticercus, and it's hard to identify on the light microscope. So I'd use one of the dissecting scopes to look at the Taenia Solium cysticercus. Now, the way people get infected is that if we eat pork that have the cysticerci in it and we don't cook the pork sufficiently, we swallow the pork with the cysticerci in them. They pass through the stomach into the intestine, and then the cysticercus, they emerge, they have hooks that they use to hook on to the inside of our intestine. They start growing and start laying eggs that we pass in our feces. Yeah, good stuff. All right. The last cast of character that you'll be looking at is a mosquito. This particular mosquito belongs to the genus Aedes. And we've included it because mosquitoes act as important vectors of microbial pathogens. We call them arthropod vectors. And again, the Aedes is so big, you really don't need a microscope to look at it. But what you can do is put it, again, under the dissecting scopes. And some students yesterday asked, why do we even look at it? You know, doesn't everybody know what a mosquito looks like? And some folks don't. You know, some folks were used to swatting them on the walls, but we've never looked at them up close. So take some time, look at the mosquito, excuse me, with the piercing mouth parts. It's got like a needle mouth part that it uses to puncture our blood vessels, and make sure that you can identify it on the lab practical and know which pathogens mosquitoes can transfer in nature. Okay. Oh my goodness, you guys, I think that's it. So we have, what, like almost an hour and a half. Oh, I knew. I was like, that was way too simple. Way too simple. We have to talk about calculating the diameter of the field of view. I know, I was like, wait a minute, we're finished.